Hi everyone. We are starting, and before Nikita will start, few words about what we will have today. So the first important thing that this is a new practice, is one of the elements of self-education. Because all of you maybe already have or will have in a few days your own scientific project. And during the work on your scientific project, of course, you will get new knowledge. And it is very important to use this knowledge and to promote this knowledge for the students. And this is one of the options, as I said, of self-education. Because if students will talk about science, it's in some cases much better than high level scientists will talk about the same topic, and you will get the knowledge from that. So that is, as I said, new practice. We did not make it before. The lectures from the students about scientific projects, about scientific work. This is general lecture about development of drug delivery systems, which is at the moment one of the hot topics in the world. So how to use drug, which is already developed, this organic molecule in most cases, and to use this organic drug to deliver into the desired target. So there are a lot of methods which we developed in the past, and Nikita will talk about them, most of them, because as I said, a lot of different methods already is being developed at the moment. And a few words about Nikita. Nikita is one of the SCAMT students, and of course he's working in my group, a group of nanopharmacy, and he really already made a good job. We published one paper in a good journal, Chem Nanomat, about magnetically controlled thrombolytic drugs, and at the moment he already has more than 3 additional projects which still in good shape, and we plan to publish all of them as well. And what else I forgot to say? Good luck. The floor is yours.
Hello everyone. Today I will be talking about basics of drug delivery and recent progress that has been made to date. And let's begin. When we talk about drug delivery, it's almost always delivery of nucleic acid-based therapeutics. And I think it's logic for you to know main types of nucleic acid-based therapeutics. So the most frequently used is small interfering RNA or siRNA, and it's kind of naturally exists in a cell, and it's used for gene downregulation. It's a rather small molecule, partly is a double-stranded molecule, and the mechanism is kind of complicated and deserves a separate lecture, so I will not talk about it. As a second type of nucleic acid-based therapeutics are optimers. Optimers are small oligonucleotides that can bind specific molecules with high affinity, and they are basically pretty much similar to antibodies. You can use it as for targeting. You can bind some receptors on some cell, or you can use it for stimuli responsive materials. Conformation of optimer is changed when it binds some molecule, and then it activates your system and the drug is released. The next type is antisense oligonucleotides. It is kind of less used because it's pretty much similar in action to siRNA, but it lacks this part of siRNA, and in mechanism it's less active than siRNA, so it's less used. Sliding again.
На поинтер направление компьютера.
компьютерной.
The next type is microinterfering RNA or mRNA. It's pretty much similar to siRNA, but it's not fully complementary to its target. So mRNA can be used for downregulation of several genes, but it has some off-target effects. Off-target means non-specific effects, so it's less used because of it. The next type is ribozymes and deoxyribozymes. My colleagues from molecular biology lab currently optimizing this system in order to work in a real cell. These are mainly artificial enzymes that produced by a method called SELEX. And these type of enzymes can cleave particular mRNA or double-stranded DNA in a particular place, like this, and the mRNA, for example, is deactivated. And the next type is matrix RNA. As you all know, matrix RNA produces protein, and you can deliver it to the cell, and it will be producing protein you need, but it's kind of large macromolecule, and it's rather hard to deliver in the cell. And the last type, the last type is coding DNA. For example, plasmid. It's extremely large molecule that can produce many proteins you need, but it's very complicated to design plasmid in order to work in a cell. So there it is. The question is why these RNAs and DNAs are most frequently used, and these nucleic acid-based therapeutics are less used. The answer is they are much smaller, they are cheaper, they are easy to decorate, easy to optimize, and these constructs are extremely large. They have relatively unpredictable action, and it's kind of complicated. The question is why siRNA is so widely used. It's more stable than other nucleic acids that I mentioned. Unlike plasmids, it needs no delivery in the nucleus. Also, in contrast to plasmids and other nucleic acid-based therapeutics, it has a much lower amount of off-target effects, and of course, it can be easily designed. The question is what issues we need to address in order to deliver siRNA in some cell you want. The main problem is siRNA has a big negative charge, and it has some difficulties while crossing the membrane, and thereby it has extremely low uptake by cells. The next problems are siRNA can trigger immune response by interacting with toll-like receptors. And the last one, siRNA is rapidly broken down by nucleases, by acidic pH, etc. How we can address these issues? Of course, we can make a complex of our nucleic acid with some positively charged molecule in order to decrease the charge. And to address the next problems, we can encapsulate this RNA in some carrier, thereby making a protective shell and providing effective endosomal escape. So, how is it difficult to deliver some? When you make an injection of your drug, first of all, it faces immune cells, it faces opsonins, it's proteins. Also, it's filtered in the liver, in the spleen and kidneys. But also, it must extravasate. It means it must go across endothelial cells of your blood vessel. It must go into extracellular matrix. It must penetrate the cell. It must escape endosome, and it must still be active. So, as you can see, pretty much complex system, and it's kind of difficult to work. First, we need to provide prolonged circulation in the bloodstream, because many of the drugs are rapidly filtered by spleen, kidney and liver. So we must control the size of your carrier. Also, we can do pegylation. I'll talk about it later. It's some modification of your system. Also, I will talk about the shape of constructs, what it must be in order to circulate long time in the bloodstream. Also, you need to control charge of your construct. Also, it must have a hydrophilic surface. It must have no barb-like modifications. I will talk about barb later. And you can coat your constructs with hydrogels. I'll talk about it later. About size control. As you can see on the left figure, it's an example from the article about gold nanoparticles. The size, the size of your nanoparticles dramatically changes by distribution of your nanoparticles. It's for 150 nanometers. As you can see, accumulation in the liver increases, and in spleen also, because when we talk about accumulation and filtration, liver can accumulate up to 200 nanometers. Also, when we talk about accumulation in the spleen, the drugs up to 1 micrometer can accumulate in spleen, and kidney can accumulate drugs up to 5 nanometers. So you can see on this example, it's DNA origami, and it's an example with DNA origami, and as you can see,
You switch on the microphone and close the door.
example with DNA origami, and as you can see, the size change of 5 millimeters dramatically changes the accumulation in liver, spleen, kidneys, on heart, lungs, and the tumor. So size pretty much matters. About regulation, it's kind of controversial thing because some articles demonstrate that regulation increases plasma concentration of your drug, and so increases circulation time. But regulation also strongly influences biodistribution of your drug, and also when you modify your construct with polyethylene glycol, it dramatically lowers uptake by cells. The thing is, polyethylene glycol forms kind of hydrogel on the surface of your construct, and it can't strongly interact with cell which you want to accumulate the drug. So the amount of polyethylene glycol in your construct influences uptake by cells. About non-spherical constructs. It's turned out that the shape of your construct influences interaction with immune cells. So when your, for example, nanoparticle faces macrophage, it can be either internalized or escape internalization by phagocyte, and it strongly depends on what shape has your nanoparticle. Also, it matters which side of your nanoparticle will be faced by immune cell. And as you can see, when the phagocyte faces nanoparticle with its larger site, its internalization velocity is dramatically decreases. So phagocyte just can't internalize these nanoparticles. As you can see on this example, when it faces nanoparticle with its small, so to say, surface, it can be internalized within 3 minutes. And when it faces macrophage with larger side of your particle, it can be internalized. What I mean, these shapes can prolong circulation in the bloodstream. There is some work with phyllomycels. It's some kind of fibers made of micelles, and they can effectively escape phagocytosis, and they can circulate in the bloodstream for about a week. The charge of your construct also influences biodistribution. As you can see on the left side, it's a negatively charged construct, it's a positively charged construct. so the accumulation statistically decreases, and as you can see, charge only matters to surface charge affects by distribution. The bigger charge, the bigger amount of interaction with serum proteins. So if your construct is having a big charge, it will be interacting with opsonins, and then it will trigger immune response, and it will not circulate in the bloodstream for a long time. Also, drug delivery systems with high surface charge are more likely to activate complement systems, or so it's related to opsonization. Also about some words about hydrophilic surface. Of course, drug delivery systems with hydrophobic surface will aggregate in the bloodstream. Also, they have poor solubility in physiological fluids. And of course, hydrophobic surface promote protein misfolding, which is not good. About part-like modifications, part or pathogen-associated recognition patterns are small parts of molecule of some invaders like microorganisms or viruses, and these small molecular moieties can be recognized by immune system, native immune system, and they can trigger immune response. These are some molecules that can trigger immune response. As I said, single-stranded RNA or double-stranded RNA can interact with toll-like receptors also and trigger immune response. About hydrogel coatings, it's some kind of polymer structure which has some strongly bound water in its structure, and it's very low immunogenic, so the immune cells poorly interact with constructs which have hydrogel coatings on it, and we can release kinetics of hydrogels, so we can release the drug in a predictable manner. Also, we can encapsulate multiple cargos, and many systems not allow this. And these hydrogels can be stimuli responsive, so you can activate it with ultrasound or light or magnetic field. When we provided prolonged circulation in the bloodstream, we must target the tissue we need. What do we need in order to do this? We can target tissue with magnetic field. We can use biolistics or gene gun. We can also use light, it's photodynamic therapy. We can use ultrasound. We can use stimuli responsive materials, and we can use EPR effect. EPR effect is related to oncology and cancer. About magnetic field. This is an example of tumor imaging using magnetic nanoparticles. It's iron oxide nanoparticles, and as you can see, while applying magnetic field, we see strong increase in fluorescence and we can actually see the tumor, while without magnetic field there is extremely low accumulation, and as you can see, some other tissues accumulate the drug. Also, as you can see on this example on the left, without applying magnetic field, vast majority of your nanoparticles accumulates in the liver, and while applying magnetic field, we can accumulate our drug in the heart. I must say some words about it because it's rather difficult to accumulate any drug in the heart because it has how to say it, it's difficult. Also, we can deliver cells to the heart using magnetic field, and the efficiency is threefold greater with magnetic field. Also, we can use a method called biolistics or gene gun. We can use micro or nanoparticles. We can coat it with our drug and just shoot it in the tissue we need, and we can, using some techniques, study the distribution of your particles, and we thereby overcoming many barriers related to the circulation in the bloodstream, the filtration. You just shoot it in the tissue you need, and you can study the how to say penetration of your particles into the tissue, and you can use just agarose gel in order to test it. You can fluorescently label it. So it's kind of interesting topic, but it's not widely used. Also, you can use light in order to activate your system. It means you inject your system, then it's biodistributes, and you can activate it in some particular place by applying light. But there is some problem with it because the depth of penetration of light depends strongly on wavelengths, and usually it's no greater than 2 millimeters. So you still need surgery in order to activate some drug. So it's called photodynamic therapy. Also, there are some works that we're using ultrasound in order to activate your drugs. As you can see, some nano-emulsions can be activated with ultrasound, and the drug is released because phase transition occurs. Also, it is some interesting work related to imaging of acute thrombosis. It is some activatable part of your drug, and it's activated by thrombin in the close proximity of the blood clot, and then nanoparticle becomes positively charged. It has some cell-penetrating peptides, alpha layer, and these molecules can accumulate in a blood clot, and you can see it using fluorescent microscope. Also, there are some stimuli responsive materials, pH responsive, ligand responsive materials. Also, you can use temperature, or you can use mechanical stimuli. Some words about APR effect. It's kind of controversial theme because it's not proven by now, but it works for nanoparticles up to 200 nanometers. So the APR effect is basically tumor vessels, blood vessels, have a larger space between epithelial cells, and we can passively accumulate nanoparticles in the tumor. Nanoparticles from 100 to 200 nanometers. So it's some kind of questionable theory. It's not proven, but it works. However, it has a modest basic targeting, so it's not very effective. When we target the tissue, we must penetrate the cell. So we can use cell-penetrating peptides. We can use receptor targeting. We can use antibodies, aptamers, and viruses. Let's talk about cell-penetrating peptides. These proteins have no receptors on the cell surface. They just positively charge. And any cell has a structure called brica-calyx. And this brica-calyx has negative charge. So it's just electrostatic force. So these proteins often contain arginine or lysine or some other positively charged amino acids, and the mechanism is pretty much simple. Of course, we can modify these cell-penetrating peptides. There are some works related to it. Also, we can deliver, as you can see, some RNA. Some words about receptor targeting. It's very much simple concept. You have some receptors on your cell which you want to target, and then you take the ligand which binds to that receptor with high affinity, and you just add it to your construct. Also, you can use so-called dual receptor targeting, so you can add receptor which binds to, you can add, ligand which binds the receptor on the cell, then it penetrates the cell by endocytosis. And you can target your nanoparticle into the mitochondria using some another ligands. So you can deliver some of your drugs into mitochondria. Of course, we can use also some types of antibodies in order to target some tissues, but it's kind of immunogenic. But there are some FDA-approved drugs based on antibodies, but it's very expensive. But you can use it. Also, we can use optimers or small oligonucleotides, which bind to some molecules with high affinity. And as you can see, it's the drug with optimer, it's without optimer, and the effectiveness increases almost threefold. And also you can use it like anti-cancer drug to target ovarian cancer. Also, you can use viruses, but there are some concerns about its safety. But you can modify it with some polymers. You can pegylate it. You can load drug which you want to deliver. Also, you can add some ligands. And it's pretty much effective system, but also it's very expensive, very expensive. Now I will talk about recent progress that has been made. I'll talk about phyllomycels, modified antibodies called nanobodies. Also, I'll say some words about main technology, cancer cell mimetics, interesting thing, and words about hydrogen logic gates. Let's talk about phyllomycels. Pretty much interesting system. As you can see, researchers added some fluorescent label to it, and you can see it in the bloodstream. And these systems are like fibers, and they have extremely high circulation time, up to 1 week, in comparison with TripToFame or stealth vesicles. And it's turned out that circulation time in the bloodstream depends strongly on the length of these phyllomycels. So, as you can see, the bigger this phylomycel is, the longer its circulation time. But when the phylomycel is bigger than 18 micrometers, it's being sheared in the bloodstream by red blood cells, so it's kind of broke down on smaller parts, so it has much smaller circulation time. Also, as you can see, there is accumulation in the liver, spleen, lung, and kidney. Also, it's a dependency of circulation time from the lens. It's a new test. Also, you can see on this picture how the conventional constructs are being internalized by phagocyte, and how phylomycels escape this phagocytosis due to its form. And some interesting thing, there are same doses of the drug. It's without phylomycels, and these are this is phylomycel 1 micrometer long with the same dose of the drug. This is the phylomycel 8 micrometers long with the same dose, and we can see a twofold increase in effectiveness. And this is eightfold increase in the dosage of your drug, and the effect is still the same. So the 8-fold increase in length of pylomicelles results in the same therapeutic effect as 8-fold increase in the dose. So it's pretty much impressive. Also about antibodies. Conventional antibodies are extremely large macromolecules, and there are some works related to its more like smallest analogs, and there are some constructs that are used. And as you can see on this figure, molecular weight of the nanobody or antibody influences renal clearance, its filtration by kidneys, and thereby molecular weight influences circulation in the bloodstream. So the smaller antibody is, the bigger is renal clearance, so the smaller is circulation time. Also, there are some difficulties with antibodies related to retargeting, because some of the receptors you target are located in the so-called caveoli. So it's kind of hole, and your antibody just can't interact with the receptor in the caveoli. So you need some linker group which elongates your antibody, and thereby it can interact with the receptor there. Also, as you can see, molecular weight of your nanobody influences accumulation in the tumor, and the bigger is molecular weight, the bigger is the amount of drug accumulated in the tumor. And as you can see, the amount of your targeted drug in the tumor is bigger when you use diabody. Diabody is diabody is here. So it's the most effective nanobody as they used. Some words about MEMT technology. This technology meant to be universal, so you can encapsulate anything, you can deliver to any tissue you want to, you can penetrate any cell you want to, but it's a very complicated system. You can complex your drug with some polymer in order to stabilize it. Then you have lipid envelope, so it's liposome. Also you have polyethylene glycol in order to prolong the circulation time, targeting ligand in order to target some cell, some receptor. Also you have cell penetrating peptides, but it's a very complicated and very expensive system, but it's still interesting to know that such a universal system exists. Also, I said about regulation, and that it is that pegylation decreases the cellular uptake, but they solve this problem by cleaving some linker, thereby polyethylene glycol is cleaved, and then your construct can simply penetrate the cell unit. So it's cleaved by some enzyme which is located in a cell you target, usually cancer cell. It's a basic mechanism of how many technology works. So the liposome goes through endocytosis, and as you can see, it is an endosomal escape. So the target drug is delivered. And as you can see, there is a glycoprotein, the part of glycocalyx, negatively charged part, which interacts with positively charged cell-penetrating peptides on the surface of MEND construct. Some words about cancer cell mimetics. It is an interesting but a very expensive way of targeting tumors. They took tumor cell, they took their membranes, they loaded with their drug, and it turned out that this construct with the cancer cell membrane strongly interacts with the same cells. So this drug very effectively accumulates in these breast cancer cells, and as you can see, it's much more effective than free drug and the drug in this capsule. So as you can see on this picture, also it's extremely high uptake of these nanoparticles by this particular stain of cells. About hydrogel logic gates, it's kind of interesting work when your hydrogel coating reacts on some stimuli. You have some proteins or enzymes in your targeted tissue, and it can interact with some parts of hydrogel. Hydrogel is broken down and the drug is released. So they have many types of logic gates, and you can tune the response of your hydrogel on some very complicated environment. As you can see, you can chemically bind the paclitax cell, doxorubicin, to your part of hydrogel, and in the presence of some enzymes, it can be broken down, the drug is released, and of course, cancer dies. There are some references. You can see through when I sent you presentation. Thank you for your attention. If you have some questions, you can say it right now, or we can talk later. Yes?
First of all, I'd like to thank you for your lecture. It was really interesting, but the main question I'm interested in is, okay, for example, if I come to a hospital in Russia and asking for help with using technology like this, would I be alive?
I think Russia is a separate thing, but I'm talking about the world trends. But Russia is developing, government is changed, so I think that is the question.
In world it is already used and
About the applicability, yeah, possible. About the applicability, it's on the stage of so some of these constructs are still in theory, some of these on preclinical studies, but it's kind of new things, and we need some times to make it in practice.
Thanks. It's really impressive to see such presentation from master students. The level is obviously higher than the master student level, but this is again a new problem arises from that. So my problem that, like, for example, I have a background in biology only from the secondary school. So I remember that the cell has a nucleus, protoplasm, and some membrane. So you send a couple of emails and also in the chat inviting us to introduce how the things can be delivered into the cell. So actually in your talk in the last part you talk about that. But what I would expect is a very simple answers to very simple questions. So what is the problem? Yeah, what is the problem exactly? So what we are talking about in problem? This is the first question. Second, if we speak about the drug delivery, how the drugs should be entered into the cell? Do we always need to break membrane? Do we break membrane mechanically or sometimes
We don't break any membranes.
We don't need to break. So okay. So this, you know, that's my questions are very simple and far from this level of representation, but still would be interesting to know.
The main questions, the main problems related to drug delivery, targeted drug delivery, are circulation times, because the drug may be filtrated rapidly and will not work in the tissue we need. And the main problem number 2 is that we need to target some particular cells and do not touch the healthy cells, and we need to separate them.
So it was some misleading when you wrote the topic like how we deliver something into the cell. That was the title, if I remember. In your email, it was written like how we deliver things into the cell. Yeah. So.
But before cell, you need to overcome some barriers.
Yeah, that's clear. And another question: if we have a tissue, I believe it consists of millions of different cells. So what is normal case? Do we deliver to every cell of the tissue, or
usually
to repair some cells in the tissue?
It's a good question. Usually the drug accumulates in the vessels close to the tissue we want to target, and some modeling works showed that it's the main problem, because when you target some tissue, you target the endothelial cells in the close proximity to the tissue, and the drug works
So it's
in that
intercellular media, yeah? Intercellular media.
extracellular.
Yeah, okay, extra, okay. So, that's it, thanks.
Yeah.
In the beginning, thank you for your lecture. And in the beginning, you were talking about the surface charge, and that the bigger the surface charge, the faster the clearance from the blood vessels.
Usually.
But we also know that the bigger the surface charge, the more stable soil nanoparticles we have. So where is this golden middle?
It depends on the system, I think. It depends on the system you are using, but you can have a lower charge simply, and that's all. And still it will be stable, but don't forget that you have a very dynamic system when you inject it into the blood, and it's kind of difficult to precipitate in the bloodstream.
Well, I think that it's easy to precipitate nanoparticles in the bloodstream.
It depends on the size.
And everything.
It depends on the size because size influences in which part of the blood vessel the nanoparticle will be, because red blood vessels in the center of the blood vessel, and they push away big nanoparticles to the periphery of the blood vessel, and it also depends on the size of your construct.
Okay. I also have a question about these phyllomycels that can stay in the organism for several weeks. Do we really need these? Such a long time?
You can design it in such a way that it will not circulate such a long time, but it's just achievement, so to say. But sometimes it matters because the drug, the thing is, the drug accumulates when it's kind of equilibrium things, and the drug must circulate many times in order to accumulate in the concentrations needed in some particular tissue.
So is it circulating or it's accumulating? Because it's different things.
Circulate.
It's circulating.
Yeah.
But how will you target it? As far as I understood, these phylomycels, they don't have any targeting.
Yeah.
So
yeah.
they can deliver this drug to any part of the body. And
The problem of this article is that it has no target. It has no target. So it's like theoretical work, and but you can modify these phylomycels with cell-penetrating peptides or some other ligands, but they didn't do it.
Okay, thank you.
Yeah.
Okay.
Any questions?
Thank you again for the lecture. I got a few questions about what is polymeric system. We got some like PEGs and these micelles. You see, I've heard somewhere, maybe I'm wrong, actually polymers can be absorbed on the surfaces and can absorb molecules, yes?
Yeah.
Am I right? So is it somehow possible that these polymers will be absorbed on the surfaces on our tissues or something and like stay
It
there?
depends on size.
Well,
Depends on size and charge, mainly charge.
So, I mean, it can happen, and it's going to be bad actually, I suppose. It's not a good idea that some materials absorbed and stays on the surface of our organism, tissues, or vessels, blood vessels, or something.
It can absorb on some surface by either hydrophobic or electrostatic forces. So it depends on charge. It's pretty much simple.
All right, I got it. And the second question, they actually can absorb some small molecules during the circulations, and can be like some mediators.
In some extent, yeah.
Yeah, so how we can control it because it's not really good, again, when we get some system that absorbs our mediators, small molecules, and so on.
We can regulate it by molecular weight of polymers you are using. Also, there are some polymers like dendrimers, so you can control the, so to say, interaction of your system with molecules in the, for example, bloodstream.
Okay, I got it. Thank you.
Thank you for a very informative lecture. I have a question. What's bigger, an antibody or bacteria?
Bacteria.
I recently read some paper which claimed that antibody is notoriously bigger.
No, it's kind of, it's, no, it's not. Bacteria is about up to 5 micrometers, if I'm not mistaken, and antibody is much smaller than any cell.
Yeah, the last question.
I remember something about antibodies, about the single chain systems of antibodies that are developed now. But it seems that they just throw away the low part of antibodies, so just we don't need it.
It's true. That's what they did.
Yeah,
But it's still very expensive because you need to generate full antibody and then cleave it.
such waste, yeah. I mean, the question is it lower part, we don't actually need it? Is it have no function or something?
The part that they are cleaving interacts with immune system. So the I think I can show it. One minute.
I know that this high part, the epitopes, and that we actually need it, but the lower part also have to have a function, I suppose.
In the antibody, we have a part which interacts with the antigen, and we have a part that interacts with immune cells in the system and activates it when antibody binds to this target antigen. So we don't need it, so we can cleave it freely.
And did I get right that by cleavage, this lower part, we just avoid immune response?
Yeah.
Okay, that's great, thank you.
So let's thank once again.